Hello everyone. My name is Samruddhi Raju Kharnar, and today I'm going to talk about the topic biomolecules, in which I have taken protein. So, what is protein? When I hear this word, I used to think about egg, meat, whey protein, gym. But is that all about protein? No, it's not. So, what's the scientific view towards the protein? So, the proteins are the molecular instruments through which the genetic information is expressed. And how does that happen? Let's understand it. So basically, genetic information is present in the form of DNA, which is present inside the nucleus of every human cell, which undergoes transcription and translation with the help of the RNA, and then the mRNA is formed, which is called as transcription in the nucleus by the enzyme RNA polymerase. Then the mRNA goes from the nucleus to the cytoplasm where the ribosomes are present, which are the protein factories. Then the tRNA comes up with the amino acids according to the anticodons or the complementary codons present on the mRNA. And then the amino acids get attached to one another by the peptide bond, leading to the formation of proteins. So what are ribosomes? So ribosomes are the manufacturer of proteins, as we discussed. It is also the protein is the major building block of the cell. And how is protein formed? It is a polymer which is made up of many monomers, which are nothing but amino acids. They can either be same or different, which are covalently bonded, and they result in the formation of polypeptides. So these polypeptides will undergo folding that can lead to the formation of primary, secondary, tertiary, and quaternary structures. Proteins are really very essential for the life to happen. Without protein, life cannot exist. What is the role of protein then? So basically, protein provides many essential functions in our body that can be in the form of digestive enzymes present in our stomach and our saliva, the antibodies which gives us immunity, lymphocytes, B lymphocytes, T lymphocytes. It also supports the regulation and expression of DNA and RNA. It also helps in the DNA repairing, replication, then supports our muscles for contraction and the movement, which is really very important for us for the mobility. Then it also provides the structure, support to the body. Hormones help coordinate the nerve signals. Then also, right now, when you're watching this video, you are breathing, and through respiration, the oxygen is being transported by the hemoglobin, which is protein inside your blood. The structure of protein, in that we'll talk about the amino acid. So what is amino acid? Basically, the amino acid has a very common structure, and it is same for all the amino acid. That is the alpha carbon atom, the amino group, and the carboxylic acid group over here. But can you see the side chain, which is the R group? That is going to vary from the amino acid to amino acid, and that's the thing which gives the different unique amino acids. Then these are the 21 different types of amino acids which are listed above, in which the amino group, alpha carbon atom, and carboxylic acid group is constant. Now let's understand how this formation of peptide bond is done in proteins. So basically, there are two amino acids, out of which the amino group and the carboxyl group are going to release out the water molecule from that. Okay, and this water molecule will do tata bye bye to that protein, and there will be the formation of peptides. What are the factors that affect the folding of protein? So the order and the arrangement of amino acids is very crucial. Then the side chains of amino acid, as I said, they are different. So they can be hydrophobic, hydrophilic, acidic, basic, that depends on that. The stability of protein will also influence the folding of protein. If it's not stable, what's the point? The stability of protein also depends on the physical factors, which are the thermodynamic and kinetic factors. Now let's understand the different structures of protein. So there are in all 4 different structures of protein which arise due to the ionic interactions, van der Waals forces, hydrogen bonds, sequences of amino acids, and disulfide interactions in the proteins which contain sulfur. So primary, secondary, tertiary, quaternary, as we discussed earlier. Let's understand what is primary structure. So primary structure is very primary. It is very simple. It is just a linear chain of amino acids bonded with the peptide bond, and it is the only form of primary protein which is going to be utilized by our body for the source of nutrition. So secondary structure. What is the second structure? First of all, the secondary structure is divided into 2 types, which is resulted due to the hydrogen bonding. It is the alpha helix and the beta sheets. Alpha helix is right-handed coil stabilized structure, whereas the beta sheet is formed due to the polypeptide chain segments, creating a zigzag or plated sheet structure. What is the tertiary structure? Tertiary structure is the 3-dimensional structure of protein, which is formed due to the interactions of hydrophobic hydrogen bonds, ionic bonds, and van der Waals forces. What are quaternary structures? These are nothing but the combination of 2 or more subunits of proteins to form a functional complex, for example, hemoglobin. Function of proteins, very simple. First is repair and maintenance of other body tissues, catalyzing chemical reactions like we saw in the digestive enzymes, and many enzymes required for replication, that is nothing but synthesizing and repairing DNA, transporting material across the cell through protein channels, giving nerve signals is what protein does, receiving and the chemical signals of the hormone, whether the hormone is required in more quantity or not required, then the cytoskeleton, which holds up the cell throughout, and the energy storage. Basically, protein is not the source of energy storage. It is just used as a building block of life. But sometimes a condition might arise due to starvation or fasting, like the carbohydrates and lipids are broken down. That is nothing but the fats. Lipids, I mean fats. So they are broken down. But what happens later? Protein is the only one remaining, but that is not a primary source of energy. But body के पास कुछ option नहीं है, तो what it will do by the process of gluconeogenesis, it will convert protein into the ATP. So it is in very rare cases, but that can happen. Now let's understand the techniques for extraction of protein. So वो होता है by disintegration of cells. The extraction of protein can be done by physical methods or chemical methods. Now it depends from cell to cell and the protein of interest, like which method you want to use. It will depend totally on that. So the chemical methods include detergents or any other chemicals which can be used for degrading the cell membrane. These are the overview of the methods mentioned in physical methods. Okay. So we are done with the extraction of protein. Now let's understand the separation techniques for proteins. So the most famous one is the SDS page. In that, let's understand its sample preparation, which contains protein buffer containing SDS and glycine, then the beta-mercaptoethanol, and a tracking dye for tracking it, and an HCl buffer. Let's understand that what is the role of each element in this. So basically, the sodium dodecyl sulfate, that is the SDS, is going to attack on the bonds that are hydrogen bonds, hydrophobic bonds, and ionic bonds, and that is going to be cleaved. Then the beta-mercaptoethanol is going to come and attack on the disulfide bond, and it will lead to the formation of protein with no interactions. Then this SDS is going to come again in the picture and get binded all over the protein molecule and giving it a net negative charge. This will lead to the formation of a linear protein. Okay, so we have converted our protein from a complex form by breaking the bonds into the structural linear form. And this is really very important to carry out its separation. Now, this is the diagrammatic representation to understand what all things are happening in the SDS page. So there are two sections in the electrophoresis of SDS, that is stacking gel and resolving gel. On the left side, we'll understand about the stacking gel. So basically, the electric field, negatively charged, positively charged, is applied towards the electrophoresis, and the stacking gel, in that, as we know in the sample, we had glycine in the buffer as well as HCl, right? So what over here is going to happen? Glycine is really very zwitterionic in nature. What it does at low pH, that is in stacking gel, it acts as a positive, that is a cation. But at a high pH comparatively in the resolving gel, it acts as an anion. Let's understand it briefly. So chlorine is the dissociated form of HCl, fluoride ions, and that are going to be leading towards the stacking gel. Why? Because the negative one is getting attracted towards the positive one. Yes. Then the glycine being positively charged is obviously going to be behind, and proteins, as you know, that SDS gave a net negative charge to the protein. So what is going to happen? This glycine and this chlorine ions are going to stack the protein inside the stacking gel, and the name itself says that. Why is it stacking gel? And then in the resolving gel, what's going to happen is it's completely ulta. The chlorine ions are in the front as they were in the stacking gel, and the glycine ions are now going to be negatively charged. Why? Because it is zwitterionic and it is a high pH. Then the protein molecules are going to follow them, and depending upon the molecular weight, the proteins are going to be separated. Okay, so this is the representation of how we can observe it by pouring out the coma C blue dye on the electrophoresis, and we can get the pattern. Now let's understand high pressure liquid chromatography. What is that? So basically, there's a liquid which is a mobile phase, that is a solvent, and there is a high pressure pump which is going to force that mobile phase into the column. It basically comprises of an injector, detector, and a computer as well. So what is going to happen? The high pressure pump will become active. It will force the solvent into the column. Along with that, the sample injector is going to pour out our sample inside that column. But how will we come to know that the sample has been poured or not? So the detector comes in the action. It will detect whether a sample is poured in it or not. And basically this column is filled with the silica, which are very smaller in size. So what will happen? The proteins are going to pass through these bead like so small silica particles, and it will get separated out based on their criteria of separation. And then we are going to detect it and lead to the formation of a chromatogram by knowing their peak values with the computer. That's it. And Obviously, as we are using high pressure pump, the column should be of stainless steel, which withstands the high pressure. And these are the different types of HPLCs, which is self-explanatory. Let's move further. So we talked about SDS field and the HPLC. So are they suitable and effective? And which is the best one? So for now, we can't comment upon which one is best among them, as it will depend on the purpose. For analytical and preparative work, HPLC is more versatile, but for routine lab checkups and molecular estimation, SDS is effective, simple, and used widely. Now let's understand about the purification techniques. What is that? Third affinity itself says that there is direct interaction of a desired molecule, which is protein, with its complementary molecular ligand that is fixed to a solid substance. And this is just basically the principle of it is same as that of the chromatography that we saw in the HPLC. Just it is कि उधर pressure apply हो रहा था, but here there's no pressure applied, and it is based on the chromatography principle and the ligand. So what will happen? The ligand will get bind to the protein of our interest, and what exactly is going to happen? The proteins which are not of our interest, because they are going to form a lock and key model, right? So which are not of our interest are going to be excluded by the mobile phase by washing them off. Okay, yes, that's the loophole. And after that, that is nothing but a buffer solution, okay, which will not interfere in the proteins ka negative positive charge. And wo hone ke baad, the proteins which are binded to that ligand, we can either change the pH or we can add a out competitor for that जो binding को वो तोड़ेगा, and then our protein can be extracted. Okay, so in this way we can get our protein in the desired form and purified form. Okay, so this is what it's happening. Okay. Salting out is really very simple. Salting out is basically used traditionally for precipitating the unwanted proteins. Yeah, sounds little tricky, but it's not. Unwanted proteins are directly precipitated with the help of salt. And over here is when we add higher concentration of salt, we decrease the interaction between the amino acid and the water, as the NaCl also wants to make bonds with the water, right? So in this way, the hydration shell, which is formed in between the amino acid and the water, that is going to be break. Okay, that is going to break down, and which will lead to the precipitation of unwanted proteins, and we can simply remove them by mechanical methods like filtration. Okay, so in order to comment on the best method for purification between these depends on our purpose. If we are aiming for high purity and specificity, affinity chromatography works best. If we are wanting to do early stage purification, we can go for salting out. Now, this is a hypothetical method, which is X-ray crystallography, which helps us visualize about the structure of the protein which we are going to study. So the purified form of protein is going to be converted into crystal, and the X-rays will hit to that protein and it will get diffracted. These diffracted X-rays are collected with the help of technology, computer, and there's a formation of electron density, which also predicts the 3D structure, and it is going to be of wavelength 1 Armstrong. Okay, the ideology of this method to form crystals is by producing supersaturated state where the concentration of proteins exceeded solubility, thus leading to the formation of crystals. So that's all for today. Thank you so much for watching my video. Please don't forget to like, share, and subscribe. Tada, bye bye.